Hello, everyone. My name is Dr. Nazish. I'm an assistant professor in the Department of Surgery, Division of Vascular Surgery, and the Stanford Cardiovascular Institute at Stanford University. And today I'd like to talk a little bit about what is the role of inflammation in aging by leveraging machine learning mechanisms as well as stem cell biology. But before I start talk, I want to give a little background about what cardiovascular aging is. It's been estimated that by the year 2030, about 20% of our population would be aged 65 and older. And even though aging presents an array of disorders, cardiovascular dysfunction or disorders has been the leading cause of morbidity and mortality among the aging population. And here I represent a slide which goes with this adage is that one is as old as its endothelium. And it's clearly quite informative because you could see that endothelial cells, which are vascular cells, plays an important role in inflammation, vascular smooth muscle cells, and proliferation, and how the endothelium or the cardiovascular system plays an important role in aging-associated disease mechanism. With that, let me start by giving a little background about the different hallmarks of aging. Very nicely represented here, we have about 9 different hallmarks of aging, from genomic instability to telomere attrition to stem cell exhaustion, which all contributes towards aging, eventually leading to many of the diseases which have mechanisms such as cardiovascular dysfunction, cancer, neurodegenerative diseases such as Alzheimer's. But one thing which we think is common among all of this is this sustained systemic inflammation, which could be contributing towards this different hallmarks of aging and eventually leading to this many different diseases which are related to aging. Now, as I talk about inflammation, this topic is about what is the role of inflammation. Let me give you a real quick background about the 2 different kinds of inflammation. One is acute inflammation, where typically, which is triggered by some sort of an infection or a trauma. It's acute, it heals much faster. You could see this kind of an inflammation if it's on your external, such as a laceration or a cut. And usually it's gone because of your response from the innate immunity from your own system. However, the one which is kind of responsible for much more of these long-term disease is chronic inflammation. And you can see it is because triggered by some kind of a physical, chemical, or a metabolic stimuli. It's a sterile agent, but it's usually undetected because of an adaptive response of the immune system, and then can lead to chronic illnesses, as highlighted here, from cardiovascular disease, which we are going to talk a little bit about, to cancer, to chronic fatigue syndrome, and thus is a little bit more undetected, dangerous form of inflammation, which could lead to many of these diseases. So based on this chronic inflammation and its involvement with the different hallmarks of aging, a new term is coined, which is known as inflammaging. It's a type of inflammation which is associated with aging and ultimately leads to some kind of a collateral damage to tissues and organs. And despite we know that chronic inflammation or these inflammatory reactions play an important role in many disease mechanisms due to aging, not many standard biomarkers are available which will allow us to identify these at a much earlier stage as they remain undetected. So we at Stanford University and at the Buck Institute of Aging set about identifying these different biomarkers which could help us identify what is the role of chronic inflammation in aging-associated cardiovascular events in our case. And for that, what we at Stanford, the Stanford 1000 Immuno project led by Dr. David Furman, they had run this longitudinal population-based study where about 1,000 individuals were recruited within Stanford and underwent a complete deep omics analysis, which included looking at their transcriptomics, their proteomics, and then passing it through a different computational modeling to kind of identify what could be the markers which are a consequence of the systemic chronic inflammation. And the major aim was to define what is the biological basis of aging. And for that, as I mentioned, about 1,000 individuals aged between 9 and 96 years old were recruited, their blood samples were collected to look at these circulating proteins, to look at their different cellular functions, and then sort of identify using machine learning what could be the potential contributors towards this aging. And really briefly, to give you an idea what was done for these different individuals which were recruited, a clinical characterization was conducted, deep immune profiling was performed so that we can then pass this information, pass this data through machine learning, and then followed it with cardiovascular assessment as we were trying to associate what is the role of chronic inflammation to age-associated cardiovascular dysfunction. So, as I said, deep learning was a very crucial component for our analysis. We had a whole omic platform being conducted, but we really wanted to pass this data from 1,000 patients to an unbiased method. And for that, what we used is a deep learning method called the guided autoencoder. And this guided autoencoder can have this data pass through so that we can then correlate what is the role of inflammation in aging. And if you look at this figure panel, you could see that we correlated the calendar age, which is the age which we have every year, compared it to something called as iAge, which is the inflammatory age. And if you can look at very clearly, this was a novel metric because we can correlate the calendar age to the iAge. And as we increase in the calendar age, as we get older, there is an increase in the inflammatory age, which is the inflammatory markers going up. There's certain outliers that you could be much younger, but have a higher inflammatory burden, or you could be much older and have a lower inflammatory burden. And this was very correlative, suggesting that as we age, we have a higher inflammatory burden. And if you can then significantly correlate between this inflammatory age and the number of disease, you could see as the higher the iAge or the inflammatory burden adjusted with the calendar age, the higher the number of diseases are usually associated with this. And more on, there is an increase in the frailty index with an increase in the inflammatory age. And you could see that there is no significant difference between the higher age and the inflammatory age, suggesting that we have a higher frailty index in these older individuals who have an increase in the inflammatory burden. So next, what we do is we set about that, let's try to identify which could be the different biomarkers, which could be the cytokine, which could be important component of the inflammatory clock. And for that, what we did is we computed this iAge, this proteomics analysis, which we had performed on these different cohorts of individuals, young and old. And you could clearly see that we found both positive and negative contributors towards the inflammatory age. You could clearly see that we found a bunch of positive contributors, which were contributing, and a bunch of negative contributors towards inflammatory age. And then when looking at the most top candidate, which was CXCL9, a cytokine which is produced in the blood, you could clearly see that there is an increase in CXCL9 biomarker in older individuals, suggesting that it has a direct correlation to the age and kind of confirming what we saw it in our computational analysis compared to the blood analysis which is performed on these individuals. Now, how do we correlate it to some kind of a cardiovascular event? We looked at two components. We looked at arterial stiffness, which is suggestive of vascular dysfunction. And clearly you could see that CXCL9 which was a positive contributor, is increased in arterial stiffness. The individuals which had arterial stiffness had CXCL9 expression on a higher level. LIF, which is a negative contributor, was downregulated. And then we looked at cardiac remodeling, which is a component of cardiovascular dysfunction. And again, you can see that CXCL9 had a higher expression in older individuals who had cardiovascular dysfunction, suggesting that now we can correlate CXCL9 to some kind of a vascular or a cardiovascular dysfunction. And this is just the same slide which we are showing, that now we can clearly indicate that our validation cohort subjected to cardiovascular assessment had a higher pulse wave velocity, which is suggestive of arterial stiffness, and looking at the right relative wall thickness, which is suggestive of cardiac remodeling. So now we can, in confidence, say that CXCL line in these older individuals who have a higher inflammatory burden is directly correlated to a cardiovascular event, which is arterial stiffness or cardiac remodeling. Now, our next goal was that can we model this inflammatory age in a dish where we can then look at this cell type, which could be responsible for an increase in the CXCL line expression. And for this, what we did is we leveraged this amazing platform originally discovered by Dr. Shinya Yamanaka, for which he won the Nobel Prize about a decade ago, where you can actually take the patient's own blood or fibroblasts and then convert them into induced pluripotent stem cells, which can then be differentiated into different cell types. And here I'm showing that if 10 cc of blood is collected, we can make these induced pluripotent stem cells, these adult stem cells, which can then be differentiated to cardiac cells, such as beating cardiomyocytes, or vascular cells, which you can clearly see a cobblestone appearance suggestive of endothelial cells. And once we have this platform, we can then model what we saw in the patients or model what we saw in our computational analysis to kind of give an organ level distinction of which cells might be producing these cytokines, especially CXCL9, contributing towards iAge. Just to show you that this platform is a very stable platform, we have used iPSCs at Stanford University to model many diseases. We have used this platform for looking at drug discovery, looking at drugs which are therapeutic, or even looking at drugs which could be cytotoxic, such as chemotherapy. And we have published a bunch of papers where we can say that this platform of induced pluripotent stem cell derived cardiac or endothelial cells is a viable platform. So, but before we kind of delve into using these iPSC platform, what we wanted to do was to look at the CXCL line from these progenitor cells. We collected primary endothelial cells from young and old individuals. Clearly, you could see that CXCL9 is much higher in the older individuals compared to the younger. But what we also did is we took the iPSC-derived endothelial cells, knocked down CXCL9, and now you can see that knockdown CXCL9 has a decreased expression of CXCL compared to the scramble, suggestive CXCL9 is secreted by endothelial cells, and an aging platform where we can age these endothelial cells derived from the same patient in a dish. So the older the endothelial cells in a dish, the higher the CXCL9 expression is. But of course, our goal was to look at a much more 30,000 feet level. Where we did RNA-seq, deep sequencing, to identify which markers could be responsible with gene pathways. And you can see, if you look at the scramble at an older passage, there is higher inflammatory response where compared to the knockdown cells, because CXCL9 is not being expressed, suggesting that CXCL9 is an important biomarker in these older individuals at a much more transcriptomic level as well. But however, we want to now go ahead and now look this at a much more granular level. So what we did is we recruited, of course, these older individuals, connected them with the RNA-seq, but then we have these older mice in our lab where we were able to age these mice up to 2 years of age, which is suggestive of older individuals. You can clearly see that young individuals had a higher relaxation. These aortas, which were isolated from older individuals, had a higher relaxation compared to older individuals, suggesting that CXCL9 is important for vascular stiffness, not only in the human patients, but also in aged mice, further validating our hypothesis that CXCL9 plays an important role in inflammation and also an important role in aging. Now, of course, we wanted to go ahead and further confirm, so we injected a CXCL9 knockdown cells in these older individuals, and you could see that there is decreased capillary formation, further suggestive of the role of endothelial cells and the role of endothelial cell senescence in aging. Overall, in this presentation, what we have shown is that the immunological fires throughout the body are causing these cardiovascular events, which could be prevented if we could identify these different biomarkers at a much earlier age. And thus, if we can identify these different markers at a much earlier stage, maybe there is a chance that we can prevent them. With that, I'll end my presentation. Please feel free to email me if you have any questions. And with that, I would like to thank everyone, and thanks for the opportunity to present my work. Bye.